Hello, and welcome back to another exciting edition on our channel. With so many adjectives one could use to define biodiversity, it is said to be the variety of living organisms which brings together different species and different forms of life. Total variety of life around Earth. The more biodiverse we are, the more secure life is. Some scientists, biodiversity is determined through genetics. Conducting research on the different genomes is key for us to comprehend the magnitude of diversity around us. Let's take a closer look and see how this can be achieved.
What is a genome? A genome is a complete set of instructions needed to create an organism. For example, your genome is a complete set of your DNA, including all of its genes. Your genes are made up of building blocks called bases or nucleotides that essentially make us who we are. Like an instruction manual, DNA can be read base by base in a process called sequencing. But why is genome sequencing so important? Genome sequencing enables scientists to read the exact order or sequence of a complete set of DNA, that is the genome, in a given organism. This provides a wealth of information and gives insight into many different organisms and can help us to better understand how they work. In another example, understanding genomes can aid in the prediction, diagnosis, treatment, and prevention of disease.
There's an infinite network where different ecosystems codepend on each other for survival, from the terrestrial to the marine life. At Inqaba biotec, we help scientists in Africa zoom into the molecular level of these life forms. Let's listen to Dr. Pepfar as he tells us more about marine biodiversity.
Hundreds of millions of people across the globe depend on the ocean for their livelihoods and for food. The basis of this is the incredible biodiversity that we found in our oceans, from the bright coral reefs through to the rich upwelling systems that we found on the west coast, bringing in nutrients from the Antarctic and supporting vast fish biomasses. This marine biodiversity is under threat from overfishing, from climate change, from pollution, plastics, etc. And we need to protect this, because protecting marine biodiversity means protecting the lives and livelihoods of people. Our work at the Oceanographic Research Institute is to try and protect and sustain marine biodiversity for future generations, not just in South Africa, but across the region and across the globe. And using modern techniques and genomics is one really important tool in our own quest to understand and to protect these vital ecosystems.
Even more exciting, and I know all scientists doing NGS-based projects are going to enjoy this. Inqaba biotec now owns the Pepfar SMRT systems, and I have a Clay to tell us more about it.
For a high-quality genome assembly, the long read is always the bet, and Pepfar's hi-fi sequencing technology will take that to the next level. This is how it works. Genomic DNA is isolated from a variety of sample types, ranging from bacteria, viruses, protozoa, you name them. The same genomic DNA is fragmented into 15 or 20 kb fragments. Pepfar adapters are then ligated to the ends of the DNA fragment, creating a circular construct. The DNA polymerase as well as the sequencing primer are then bound to the adapters, creating what we call a SMRT Bell DNA polymerase complex, which is then loaded onto the SMRT cell for sequencing. At the heart of the SMRT technology is the SMRT cell, which contains millions and millions of tiny wells known as the zero mode waveguides, or ZMW in short. The DNA polymerase, the SMRT Bell library complex, is bound to the bottom of each and every ZMW. As the DNA polymerase incorporates fluorescently labeled DNTPs A, C, T, or G, the light is emitted and captured in real time, as we now wait for the system to complete for secondary analysis to take place bioinformatically.
I hope you find a short review on the Pepfar SMRT system as fascinating as we do. Thanks, Clay. Now, Inqaba is offering you a chance to be part of our own history by engaging in the Inqaba's African Genome Challenge. We offer you an opportunity to sequence up to 3 individual samples of the same species at absolutely no charge. Details on how to join in these competitions are on the description box below. All subsequent updates will be available on our social media platforms. And we are not leaving out our non-scientist friends. You too can be part of this challenge by choosing the projects that you feel are the best to win in this competition. All details will be shared on the description box below. Until next time, goodbye.